This is sexual reproduction with meiosis from chapter 10 from the Campbell's Biology, AP Biology in Focus. The importance of sexual reproduction and meiosis is that it causes variation. Variation that the natural selection can work on it, and as the natural selection will drive the evolution. Genetics is the studies of the transmission of traits from one generation to another, as well as the variation in phenotypes in a population. The reproduction that we have to just go very general about that can be asexual reproduction when as well as sexual reproduction. With asexual reproduction, there's one parent and it's going to have two offsprings, and the offsprings are identical to each other as well as to the parents. No sex cells are required or gametes for this process. The identical DNA for all three of them. This is very energy efficient process. They don't have to find mates, and it's a fast process. Example for that or the types are budding, fragmentation, regeneration, and binary fission. The other type of reproduction is sexual reproduction, when we have two parents that they are genetically different, slightly different from each other, and their gametes fuse together to form a zygote, and the zygote contains different genetic material, the combination of the parents' genetic material. It works with gametes, sex cells, and provides genetic variation. It is evolutionary advantageous, although the process is slower and requires more energy in order to find mates too and produce these sex cells. But the natural selection can act on the species, and only the fittest would survive, and the species will hopefully will not die out. Focusing on the humans now. In the humans, there are two types of cells: the autosomal cells, the somatic cells, as well as the gametes. Autosomal cells are the somatic cells. Soma means body. So it's my heart cells, muscle cells, skin cells. They all contain 46 chromosomes or 23 pairs of chromosomes. The cells are diploid, and we shorthand write that 2n equals 46. Gametes are the sex cells. They are produced in ovaries and the testes, or the testes. The ovaries product is the egg, and the testes product is the sperm. When the two egg and the sperm joins together, they will the sperm fertilizes the egg, and they form the zygote. The zygote is a diploid cell, and the zygote will go through meiosis that will form the organism. The egg and the sperm formation happens through meiosis or meiosis. The egg contains 23 chromosomes as well as the sperms, and the zygote will have 46 chromosomes. The cells and the chromosomes. Let's assume we have a cell, and the red will represent the maternal genetic material, and the blue is the paternal one. This cell is a diploid cell, contains 4 chromosomes. or yeah, those are homologous chromosomes, and that means that their shape and the genes loci are similar on both of them. Now, this cell is going to go through cell division, so it has to go through the S phase when the genetic material will duplicate. So we will have duplicated chromosomes. Also, the amount of chromosomes is 4. So these in the middle, these are the centromeres, and they are attached to each other by cohesion. Now the karyotype, what we will use in class, usually made in metaphase, and in humans we will have 22 pair of autosomal chromosomes. They are duplicated, and 1 pair of sex chromosomes. For females it contains 2 X's, and for the males it contains 1 X and 1 Y chromosome. And the males, oh, never mind. The meiosis or meiosis. Now, the parental cell, for example, this will contain 2 pairs of chromosomes. Parental cell contains 2N, is 4, and this is an oocyte or a spermatogonium. This is the diploid cell that will give rise of haploid cells. After the S phase, the chromosomes, the genetic material will duplicate. So I have 4 paired chromosomes, 4 duplicated chromosomes, or 2 pairs of duplicated chromosomes. What I circled is the tetrad homologous pairs. Tetrad will be important in that process. And the first section of meiosis is meiosis 1 or meiosis 1. This is a reduction division when a diploid cell will turn into a haploid cell, 2 haploid cells. We have the cells, and the homologous chromosomes separated. So from the big chromosomes, one duplicated will go to the left, and another duplicated will be on the right. At prophase 1, crossing over will take place between homologous chromosomes, but not between the sister chromatids. At the chiasma is when the 2 homologous chromosomes cross over. Metaphase, during metaphase, the chromosomes line up in the metaphase plate, and microtubules from the centrioles, from the centrosomes, start to connect with the kinetochores, and they arrange them in the metaphase plate. Anaphase 1 is when the homologous chromosomes separate, but the sisters stay together. Telophase is when the haploid set with the sister chromosomes will be in each half of the cell. And the cytokinesis, the 2 haploid cells will form cleavage furrow with animal cells and cell plate with plant cells. The other part of the following part of meiosis is meiosis 2, when it's an equal division. Haploid cell will turn into another haploid cell, 2 haploid cells. So we will have altogether 4 different cells. Now they are similar because we didn't play through the crossing over, but meiosis 2 is very similar to mitosis. It also has the prophase 2, when the spindle fibers form towards the metaphase plate. Metaphase 2 is when the sister chromatids line up at the plates. At that point, they are not identical due to the crossing over. And the kinetochores kinetochores get attached. The microtubules get attached to kinetochores, and a phase separating the sister chromatids from each other. They act as individual chromosomes. And telophase 2, when the nuclei start to form and decondensation of the chromosomes happen. We don't still have 2 cells. The cytokinesis has to kick in. So we will have 4 genetically different cells at the end of meiosis. This is the bottom line. The cell division, getting back to cell division, we will compare mitosis and meiosis. Mitosis, we have covered it earlier a little bit. But in here, genetically identical, the parent is genetically identical to the daughter cells. We will have a diploid cell that gives rise to another diploid cell, or haploid cell to haploid cell in plants or in fungus, and it happens once the division. On the other hand, meiosis produce haploid cells from diploid cell. Parent and daughter cells are genetically different, and the daughters from daughters are different too. It contains 2 divisions, and crossing over happens at the synapses. It's only happening in meiosis, nowhere else. Then the homologous chromosomes line up next to each other to form tetrade. Both of them has cohesins at the centromere that breaks at the end of metaphase or the beginning of anaphase. The cohesin cleaves at the beginning of anaphase 1 between the homologous pairs to break up the tetrad, and at the beginning of anaphase 2 between the sister chromatids. Variation in the species come from independent assortment, when homologous chromosomes orient randomly at metaphase 1. It also comes from the crossing over between homologous chromosomes, but not between sister chromatids, and it comes from random fertilization. And that's all.